All right, hello everybody. This is Miss P, and I am here today to kind of talk about what we're going to be learning about this week and in the coming weeks. So basically, what we're going to focus on for the first few weeks of our distance learning experience is DNA. We're going to focus specifically on DNA, like what it is, how it copies itself, all of these interesting things. But for this week specifically, we are going to be focusing on what DNA is in the first place. And so I've got a couple learning goals for you guys. The first one is that I want you guys to be able to model and describe the structure of DNA by the end of the week. This can look different for different people. There'll be opportunities to make a 3D model of DNA, but you can also do drawings or other things like that. Just let me know which way you want to model it once you get to that task, and we can talk about it then. A second goal is that I want you to be able to discuss your opinions about the discovery of DNA. So there's actually a little bit of an ethical dilemma surrounding the discovery of DNA. So that means that there are some opinions out there that believe that the people who get the most credit for discovering DNA shouldn't actually have gotten all that credit. And I want you to be able to discuss your opinions of that situation, whether they're for one side or the other. The opinion itself doesn't matter. More so, I'm looking for how you express your opinion. Can you express yourself in a very eloquent and good way? Can you talk about why you believe what you believe, basically speaking? So these are our two learning goals today. I want to apologize in advance for our interesting seating chart that we've got going on right now. This is not what I meant when I said I was giving you guys a new seating chart after your last exam. I didn't think we'd be this far apart. But that said, we're going to do our best in the situation that we have, and we're going to learn as best as we can given the information and given the resources that we have. So I'll be making videos like this approximately once a week for you guys to tune in and basically hear me talk for a little bit. Maybe in the future I'll put my face on the video, but I would not expect that from these. So with that said, let's begin. Okay. So, just to give you guys a little bit of background about how in the world we got to this point. Right before our break, before our spring break, we were talking about genetics. Specifically, we were focusing in on something called Mendelian genetics. So those are genetics where we're thinking about specifically traits and characteristics. So remember we did that exercise with the cooties. We had a reddish colored cootie and a bluish colored cootie, and we bred them together and we did some Punnett squares to figure out exactly the likelihood that they would have certain color offspring. So that focused on genotypes and phenotypes, right? The genotypes were the letters inside the Punnett squares, and the phenotypes are what the organisms actually looked like. So down here, we would see the phenotypes. In the Punnett square, we would see the genotypes. That last week, the week and a half before spring break, we were looking at pedigrees. Those are also something that are considered a part of Mendelian genetics. But we are going to be done with those now. You've taken your quiz on them. If you haven't, please check out Canvas. There is an opportunity for you to take your quiz on Canvas. You can contact me later if you have questions about that. But for now, from now on, we are going to be moving forward into something else called molecular genetics. So at this point, instead of focusing on the traits and characteristics of organisms, we're going to be looking at something a little bit smaller than that. So we're going to be looking at things like DNA, RNA, and proteins, and how those things inside our bodies and inside our cells actually make these traits and characteristics happen. So we're going to learn about DNA, we're going to learn about RNA, we're going to learn a little bit about proteins, and we're going to discover, basically, how these different structures in our bodies make us who we are. So, what is DNA in the first place? Most of us might be able to say that DNA is the stuff inside our bodies that makes us who we are, right? But for our purpose, we're going to be thinking about DNA in a slightly new way, a little bit more in-depth way than we might have in the past. So DNA is the chemical that is the genetic code for all living things. So if you find a living thing anywhere, It has DNA in it somewhere. The presence of DNA is one of those things that symbolizes to scientists and to people studying these things that that thing actually is alive. Not all things with DNA are alive, like viruses have DNA, but we don't consider them alive. So think about the coronavirus. It technically has viral DNA, but it's not alive. It can't reproduce without a host, like us, which is why we're so far apart right now. But nonetheless, DNA is that genetic code for all living things. It's a chemical, and it's like official chemical name if you want to say it in fancy terms or scientific terms. DNA stands for deoxyribonucleic acid. And these terms are just very, very fancy scientific words for what DNA is. You won't be tested on the actual name of DNA. It's focused on down here, but I will want you to know what the basic definition of DNA is, which can be found up here. So what is DNA made up of? Like, what makes it what it is, basically speaking? DNA is made up of 3 other smaller molecules, basically speaking, smaller parts that make up the whole. The first part, or substance that is found inside DNA is a 5-carbon sugar. So that's going to be kind of like right here on this smaller little piece. You can see this sugar has 5 points. Each of those points would have a carbon atom. You don't need to know super specifics. I just want you to know that it's made of 3 other molecules, and I would like you to be able to describe those 3 molecules. So the first one is a 5-carbon sugar. The second molecule that makes up DNA is a phosphate group, kind of over here. And the third is a nitrogen base or a nitrogenous base. You'll hear me refer to them in both ways throughout this presentation. They are the same thing. Nitrogen base, nitrogen base are the same thing. They are over here. So when these 3 molecules combine, they come together to form something called a nucleotide. So a nucleotide is kind of that building block of DNA. Back in December, when I first started showing up in the classroom, you guys learned about nucleotides. You learned that nucleotides were the things that make up DNA. So kind of if you're thinking about DNA as a puzzle, nucleotide would be the puzzle pieces. If you're thinking about DNA as a ladder, it could be like the rungs on a ladder. Each rung is a nucleotide. So nucleotides are those building blocks of DNA. There are a few different kinds of nucleotides that we can find in DNA. For our sake, we're going to say there are 4 different nucleotides. There are 4 different nucleotides in DNA. And these nucleotides differ based on the nitrogenous base that they have. So there's 4 different kinds of nitrogenous bases that can be attached to that 5-carbon sugar and that phosphate group. And those 4 nitrogenous bases are adenine, symbolized by an A, guanine, symbolized by a G, thymine, symbolized by a T, and cytosine, with a C. I won't ask you to be able to draw these nucleotides, so don't worry about paying attention to like the specific little things hanging off of all of these nucleotides. I just want you to know that there are different nitrogen bases, and that those nitrogen bases are what make different nucleotides different. As I said before, these nucleotides are those building blocks of DNA. So they can link up and form long chains, and those chains, those polymer chains, those many nucleotide chains, kind of form what DNA is. And they link up in a very specific way. And it happens when the sugar and phosphate portions of the nucleotides bond together. So you can see over here, we have our 5-carbon sugar and our phosphate group, and those pieces of the nucleotides are linking up in a chain. They're lining up in a chain. Ooh, excuse me. This chain of sugar and phosphate molecules is called the sugar-phosphate backbone of DNA. So if we were thinking about it and you wanted to individually see the nucleotides, you could think about it in this way. Here's one nucleotide, here's a second nucleotide, third nucleotide, a fourth nucleotide, and a fifth nucleotide. And they're all linked by that sugar phosphate backbone. Over here, I have another image of DNA. On this, the sugar phosphate backbone is symbolized by this purple ribbon-like border. You can kind of see it is a backbone for the shape of the DNA, which we're going to talk about very soon. DNA isn't just one strand of nucleotides, though. DNA is a double-stranded molecule, which means there are two chains of nucleotides for any given piece of DNA. You'll see over here we have one sugar phosphate background over here and another sugar phosphate backbone over here, which means there are two chains of nucleotides for every piece of DNA. We call the shape that these double-stranded molecules form, so they don't, a double helix. A double helix looks like this molecule over here. If you want to think about it in ordinary terms, this shape kind of looks like a twisted ladder. So if you took just like any old ladder, any ladder that you would use to reach something when you're painting or cleaning in your house, and you twisted it, it would kind of look something like this, right? This is the actual shape of DNA. So that double-stranded double helix, a double-stranded molecule that forms a helix or kind of like a swirl, if you will. To make this double helix happen, those two strands or those two chains of nucleotides have to get together somehow. They combine and they link together by having their nitrogen bases pair up. And this happens in a very specific way for DNA, and it happens the same way in all organisms. All living things, all DNA that's ever been analyzed, ever been looked at, certain bases always pair up with other bases. For example, adenine, or A, always pairs with thymine, or T. And cytosine, C, always pairs with guanine, G. So you can see over here on our image right here, it's just kind of a flat 2D image of what DNA could look like. We have our thymine nitrogen base paired with our adenine nitrogen base. Here we have an adenine paired with a thymine, a guanine with a cytosine, another thymine with an adenine, and another guanine with another cytosine. Notice it doesn't matter which side the G and the C and the A and the T are on, but they always pair up with the same molecule. So you'll always find the same base opposite another. T will always be across from A, regardless of which side of the DNA strand it's on. All right, at this point, I want to give you a chance to kind of test yourself. You've been listening to me talk for quite a while now. I want you to pause the video and look at this DNA code right here. Now you know that certain bases or certain letters always pair with other letters. So if this is one side of that double-stranded DNA molecule, what does that second strand look like? I want you to take a minute, maybe find a piece of scratch paper or a pencil or a pen or something, and I want you to write down what you think the complementary strand to this one would be. So what would be found on the other side of this DNA molecule? So if like this sequence was right here, what letters would you find right here? Does that make sense? Okay, take a couple minutes, pause the video, go for that. All right, welcome back. I hope you took a couple minutes and thought about this DNA strand and what the other strand, that complementary strand to this one, might look like. Hopefully by now you've paused the video, you've taken a minute to write that down so you have it in your brain. Test yourself. All right. Here is what that second strand would look like. So you can see it down here toward the bottom. your first strand would have ATG, GCG, and TGA. That's just one half of the DNA strand. There's a second half. That second half, that complementary side to the first strand, would have certain bases pairing with others. So we know that thymine, or T, always pairs with A. So the first letter on our second strand should be a T, and it is. We know that thymine always pairs with adenine, so we should see an A on the second strand, and we do. Likewise, guanine always pairs with cytosine. I want you to take a minute and compare the second strand to what you wrote down for your second strand, your complementary strand. Did you get it right? Do you understand why or where you have a problem if you made a mistake? If you want more practice with this, please pop into office hours. I would be glad to give you a practice question. All right. There's a couple more things about the structure of DNA that I want you to be able to recognize or talk about or explain. I want you to look specifically at the sugar phosphate backbone in this picture. In a double helix, so in a molecule of DNA, in a piece of DNA, those backbones, the sugar phosphate backbones, are anti-parallel, which basically means that they run in opposite directions. And we can look at the structure of DNA and we can label those ends with certain names. One end is going to be a 5' end, or 5 with a little apostrophe end. We call that 5' end. That end is going to be one where the phosphate group is sticking out unbonded. That is how we identify a 5' end. The other end of that strand is called the 3' end. That end is the end where there is no phosphate group. It's just the 5-carbon sugar hanging out. So we like to think about strands of DNA as running from 5' to 3'. But if you look at the other strand, the complementary strand, the second strand of this double-sided molecule, this double-stranded DNA, this one runs from 5', we can see down here we've got an open phosphate group, to 3', where we've got that sugar group at the end. So in this way, we can see that these two strands run in opposite directions. 5' to 3' over here, but over here it would be 3 to 5. This is what I mean when I say that DNA strands are anti-parallel. They run in opposite directions. So that's all like great, Miss P, right? Like, okay, now I know way more about the structure of DNA than I ever thought I wanted to and that I ever want to remember again. So I will, later in the week, be asking you to talk about the structure of DNA. And I'll give you some guided questions to help you along your way. That's in a later task. You don't have to worry about that at this point. This is just to get you started and to be a resource for you to come back and check when you're making your explanation. But there's a little bit of history behind the structure of DNA too. So I want you to think about who discovered the structure of DNA. Who was the scientist or the group of scientists that figured all of this stuff out, and how in the world did they do it? So the short answer is that many scientists contributed to our current understanding of DNA. Many scientists over many years put their life's work toward figuring out the structure of DNA. So they figured out the 3 parts that we talked about. They figured out how those nitrogenous bases pair together. They figured out that those nitrogen bases, that A, T, C, and G, they figured out they were different in the first place. But it took a while for a group of scientists to actually put all that information together and to come up with this double helix structure that we've been talking about for the last few minutes. So for the most part, credit for this structure goes to 2 men. Francis Crick and James Watson. In 1953, they published a journal article in which they talked about a hypothetical structure for DNA. And based on what they had found by analyzing everybody else's research who had worked on DNA up to that point, they figured out this double helix structure, and they kind of built a model to help explain that structure. So this is actually a photo of them, of Crick and Watson, kind of explaining their structure in a way. They are the people that a lot of us think of when we think about DNA. We think, oh, DNA, that structure was discovered by Crick and Watson. However, they were not the only scientists on the job. There were a bunch of other scientists also working, or racing, if you will, to figure out the structure of DNA. Two of them were Maurice Wilkins and Rosalind Franklin. Both Wilkins and Franklin separately published articles about the structure of DNA in the same journal issue about the same structure for DNA. So these two scientists also figured out the same structure for DNA at the same time Watson and Crick did, but they don't often get as much credit. Watson, Crick, and Wilkins eventually earned a Nobel Prize for their discovery of the structure of DNA in 1962. Rosalind Franklin was ineligible to win a Nobel Prize for her contributions to the structure of DNA because she had died of cancer in 1958, and Nobel Prizes can only be awarded to living scientists. In a future task, you're going to learn a little bit more about Rosalind Franklin. She was a woman scientist in a time where science was dominated by men. And a lot of people think that she should get more credit for the work she contributed to the structure of DNA than she actually does. You'll learn a little bit more about that in the future, and that is where the controversy comes into this. So I don't want to reveal too much. You'll have plenty of time in another task to take a look at it. But for now, that is all I have for you this week. I hope you were able to follow what I was presenting. Feel free to rewatch this as many times as you need. Take your own notes if you think that that will help you when you're explaining down the line. And have a great week. I look forward to hearing from all of you.